We're diving deep into another important immunohistochemistry test, the INI-1 test. This test is crucial for diagnosing certain types of cancers, and understanding its significance can provide valuable insights for both medical professionals and patients. So, let's get started. Pre-test information. Before we discuss the specifics of the INI-1 test, let's talk about what you need to prepare before the test. First, you must provide a copy of the histopathology report. This report offers a detailed examination of the biopsy sample under a microscope and is essential for interpreting the INI-1 test results. Next, it's important to provide the site of the biopsy. Knowing the exact location from where the tissue was taken helps pathologists understand the context of the sample. Lastly, the clinical history of the patient should be shared. This includes any relevant medical history or symptoms that might relate to the disease under investigation. Specification of the INI-1 test. The INI-1 test is a specialized immunohistochemistry test that helps detect the presence or absence of INI-1 protein in tissue samples. INI-1, also known as SMARCB1, is a tumor suppressor gene that is expressed by most normal cells in the body. The test uses specific antibodies to determine if the INI-1 protein is present or has been lost, which can be a critical indicator of certain malignancies. Diseases associated with INI-1. So, what diseases are associated with the loss of INI-1 expression? Loss of INI-1 expression due to mutations is most commonly seen in malignant rhabdoid tumors and epithelioid sarcomas. These are aggressive tumors that often require early and accurate diagnosis for better treatment outcomes. Additionally, studies have shown that some glioblastomas, a type of brain tumor, also exhibit loss of INI-1 expression. This suggests an underlying molecular aberration, leading to a more aggressive clinical behavior in these cases. Specimen used in the test. For the INI-1 test, the specimen typically used is a tissue sample obtained from a biopsy. This could be from any tissue suspected of harboring a tumor. The quality and preservation of the sample are crucial for the accuracy of the test results. Components included in the test. The INI-1 test involves several key components. Primary antibody. This is specific to the INI-1 protein and binds to it if present in the sample. Secondary antibody. This binds to the primary antibody and usually has a marker enzyme attached to it, which helps in visualizing the results under a microscope. Detection system. This involves using a substrate that reacts with the enzyme to produce a color indicating the presence of the INI-1 protein. Test report time. Once the biopsy is taken and the test is performed, the report time for the INI-1 test is typically about 7 days. This includes the time required for processing the tissue, performing the test, and interpreting the results by a pathologist. Test information and conclusion. To sum it up, the INI-1 test is a vital tool in the diagnosis and management of certain cancers. By detecting the loss of INI-1 expression, pathologists can identify aggressive tumors like malignant rhabdoid tumors, epithelioid sarcomas, and some glioblastomas. This knowledge is crucial for guiding treatment decisions and improving patient outcomes.